everyone, welcome back to my channel Study More. I am Mousumi and today I am going to teach you about animal cell, and this is the English version of this video. So I have already uploaded the Hindi version of this video. If you want to watch that, you can easily go to my playlist and you can find that video there. Okay, so let me start. So I have taken a piece of paper where I am going to draw the outline of the cell. Okay, so first of all you have to draw this kind of line, and then after that a single straight line, and again you will make a cut like this. Then you will make foldings at the top. After making this foldings, again you will draw a cut, and then you have to draw the line, and then you come to the bottom portion, and similarly you will create some cuts at the bottom portion also, and then try to connect this two lines. Okay, so as you can see, the outer part of the cell is already created. Okay, so this outer part of the cell is known as what? Do you know that? This is known as cell membrane, and the other name of the cell membrane is plasma membrane, or we can say plasma lemma, or it is also known as cytoplasmic membrane. Okay, so you have seen already that there are two cuts present over here. So these cuts are actually meant for the process of endocytosis and exocytosis. That is, if any substances wants to go outside the cell, then with the help of this portion they can easily go outside the cell, and if any substance wants to enter the cell, then that is also done through this portion. I hope it is clear to you. Now let me tell you one thing, that this cell membrane was actually discovered by Swiss botanists in 1855. Okay, now in order to remember the name of the two botanists, I want to tell you that from the word cell you get a C word, isn't it? So with that C word, the name of the scientist is already will get, that is Kramer, and another name is Nageli, and this Nageli starts with the letter N, and just after a membrane, that is after letter M, comes the letter N. So in this way you can easily remember these two names, that is Carl and Nageli. And as you can see that at the top the foldings are there. Can you see that? Yes, I'm talking about this foldings. The main function of this foldings is to create what you know. Actually it helps to increase the surface area. Okay, so it helps to increase the surface area. Now let me draw the nucleus. Okay, so I will draw the nucleus. So you will draw in this way. It is a double membrane, so leaving some gaps you will complete this nucleus. Okay, so in this way you have to draw the nuclear membrane. Okay, and in between this there is a pore, which I will discuss later on, but let me level that at first. So this is known as what? This is known as nuclear membrane, or you can say nuclear envelope. Okay, so this is the outer covering of the nucleus, and as you can see, can you see that in between the nuclear membrane there are some gaps, or there are some pore. So this is known as what? Do you know? This is known as nuclear pore. So let me level that also. This pore is known as nuclear pore. Now you can ask that what is the main function of this nuclear pore? Actually it acts as the connection between the cytoplasm and the nucleus. Okay, if anything wants to come inside this nucleus from the cytoplasm, or if anything wants to go outside the means nucleus, so with the help of this nuclear pore, these substances can easily do that. I hope it is clear to you. Now let me draw the next structure. Okay, I will draw the next structure now. The next structure is what you know, that is nucleolus. Okay, let me use another color, and with the help of that color I will draw this nucleolus. It is very dense body which is present inside the nucleus. Let me level that also. And what is the main function of this nucleolus? Actually, this nucleolus helps in the synthesis of ribosomes. Okay. Ribosomes I will discuss afterwards, means now only I will discuss, but before that I want to tell you that it helps in the synthesis of ribosomes, and there are some materials which are present beside this nucleus that which contains DNA and proteins, and this is known as, what do you know? This is known as chromatin material. I hope it is clear to you. So this one is chromatin material. I've got to level that nucleolus portion. So let me level that portion also. So this one is nucleolus. So after that we can easily see that all the 4 parts of the nucleus I have already labeled. So nucleus is made up of all these 4 parts. Now I will draw the endoplasmic reticulum. Okay, so this 4 parts of the nucleus I have already drawn, as you can see. So let me start with the endoplasmic reticulum. So in this way you have to draw, just concentrate on the diagram how I am doing it. So one in this way continuous thing you have to make, and this type of structure should be present all around the nucleus. Okay, these are actually you have to make in such a way that it surrounds the nucleus in all the portion. Okay, just like this you have to make it. I hope it is clear to you, because endoplasmic reticulum is one of the most important organelle which is present inside the animal cell. So in this way you have to draw the diagram. Okay, so I have already drawn that. Just see clearly how is it looking. So in this way you have to draw the diagram of the endoplasmic reticulum. Now let me do the next organelle, that is ribosomes. I will do that with a different color. So just see, just above the endoplasmic reticulum you will see some ribosomes present, or some ribosomes are attached to this endoplasmic reticulum, but don't think that always these ribosomes are attached to this endoplasmic reticulum. But the ribosomes which are attached to the endoplasmic reticulum, when it is attached, then that is known as RER. Okay, that is rough endoplasmic reticulum. Now here I am drawing the endoplasmic reticulum which is devoid of these ribosomes, and whenever the endoplasmic reticulum is devoid of ribosome, then that is known as smooth endoplasmic reticulum. I am writing in short, that is SER, but actually it means smooth endoplasmic reticulum. So as you can see, when ribosomes are attached to the endoplasmic reticulum, this type of ribosomes are completely different to that of the endoplasmic reticulum where the ribosomes are not present, and some ribosomes are freely present inside the cell. As I am drawing here, you can easily see that. So these are known as, what you know, these are known as free ribosomes. Okay, so let me label that. This is known as free ribosomes. Okay, now these free ribosomes and this ribosomes which are attached to the endoplasmic reticulum, that is known as ER bound ribosomes. Okay, so ER bound ribosomes or endoplasmic reticulum bound ribosomes. Now the main difference between this endoplasmic reticulum bound ribosomes is that it creates the proteins which are required outside the cell, and the free ribosomes make the proteins which remain inside the cell. Okay, this is the main difference between the free ribosomes and the ER bound ribosomes. This you have to keep it in your mind. Okay, that difference is very, very important. So two types of ribosomes will generally found. Now let me draw the Golgi apparatus, one of the important organelle in the cell membrane. You have to draw in this way, smaller to larger one. This is also actually, this is actually a double membrane to this one also, and the other name of Golgi apparatus is Golgi body, or we can also say Golgi complex, or we can also say dictyosome. Okay, now can you see the budding which I am actually making at the top? So you can also do it, and actually it helps in the production of certain vesicles or certain organelle that I will discuss. That this Golgi apparatus already have leveled. Now this bud which I have created, it creates certain organelles, that is lysosomes. Okay, the name of this organelle is lysosomes. Now about lysosome obviously I'll discuss, but let me level that one also. I hope it is clear to you. Okay, now first let me tell you the function of the Golgi body. First of all, Golgi body actually processes the protein which are actually synthesized by the ribosomes. Protein processing and packaging for sending it outside the cell. Okay, now I am making the transport vesicles. As you can see, the transport vesicles which help in the transportation. So whenever there is any protein transportation, it is actually managed by this Golgi body only, and the transportation is done by the transport vesicles. So the Golgi body actually decides which protein will stay inside the cell and which will go outside the cell. This portion you have to remember. And Golgi body also produces the lysosomes. Okay, it also produces the lysosomes. And one thing I want to tell you, that this transport vesicles help to transport the proteins. That you have to remember. Okay, and now I will discuss about this lysosome. So let me take another page, and here I am going to discuss about the word lysosome. Now lysosome is actually made up of two portions. Now this word lyso, this part lyso means lysis, okay, and soma means soma, or a body. Clear? Now lysis, what is the meaning of lysis? Lysis means break down or digest. If anything has to be broken down or for digesting, this word lysis is being used. So obviously some digestive enzymes are present inside this organelle, that is inside the lysosomes. Certain digestive enzymes are present, and this digestive enzymes are produced where? These digestive enzymes are actually produced by another organelle, and the name of this organelle is rough endoplasmic reticulum, that is RER. So it is produced by RER. Now whenever this digestive enzyme is produced by RER, this should be transported by something. Okay, otherwise how will it reach to the lysosome? So this is being actually, whenever it is produced by the RER, it is being transported by the transport vesicles, and these transport vesicles carry this to the lysosomes. So let me write it down, that this digestive enzymes are actually transported by the transport vesicles. So these are transported by the transport vesicles. Now I will show this in the diagram. So whatever I have drawn, so it will be easier for you to understand. So let me bring the previous paper once again. So I have brought that. Now just see this one, that whatever enzyme is produced, that is the digestive enzymes are produced by the ribosomes, okay, which are present or which are attached to the endoplasmic reticulum. And when these digestive enzymes are produced, these are transported by these transport vesicles which I have marked. and this transport vesicles again take this or transport this to the lysosomes. Okay, I hope it is clear to you. So in this way the thing is being transported. Now I will discuss about the next portion, that is how to maintain the cell structure. So to maintain the cell structure, actually three things are needed. Okay, three things are actually present inside the cell which helps in the maintaining the cell structure. So first one is a microtubule. Okay, microtubule, or you can say that these are very small tube like structure, we can say solid tube like structure, which helps to maintain the structure of the cell. Okay, now the thing is that the next one I will tell you that the next portion or the next part which helps to maintain the structure is the microfilament. Okay, these microfilaments are actually thinner than the microtubules, and this also helps in maintaining the structure of the cell, which I will show you in the diagram also. Okay, now the third portion, let me tell you that one liquid is present, and the name of the liquid is cytosol. Okay, cytosol, and it is liquided, it is in liquid form, and it helps to tighten the cell mainly. And the word cyto means cell and sol means solution. So cytosol is made up of these two words or two parts. Cyto, that is cell, and sol means solution. Okay, so whatever liquid is present, that is known as cytosol. Now cytosol plus the organelles which are present, and if we consider this, then that is known as cytoplasm. I hope it is clear to you. So cytoplasm is actually, if we deduct the plasma membrane and if we do not consider the nucleus, then we will get the cytoplasm. Okay, so without considering the plasma membrane and the nucleus, if we only consider the cytosol and the organelles above this cytosol, then that is known as cytoplasm. Okay, so now again let me bring the diagram which I was using. So let me bring that paper once again for you. So just see the diagram which I have drawn. So first I will draw this microtubule. Okay, so this one is microtubule, which I have already discussed that it helps to maintain the structure. Now with a different color I will draw the microfilaments. So these are the microfilaments which you can see. These are thinner than the microtubules. Now let me mark that also, that this one is microfilament. Okay, so this one is microfilament, and now I will mark the microtubule also. So I've got to actually mark this or label this microtubule. So let me label that microtubule, and then as you can see that the liquid portion, that is known as what? That the liquid portion is known as cytosol. Okay, I've already marked the microtubule and the microfilament. So this is the plasma membrane and the nucleus which I am showing. So without this plasma membrane and the nucleus, the remaining portion without the organelles, that is known as what? Without the organelles I'm talking about, that is known as what? Cytosol. And if we consider the liquid portion, that is the cytosol, and also the Golgi apparatus, Endoplasmic Reticulum, ribosomes, lysosomes, that is all the organelles if we consider, then that is known as what? That is known as cytoplasm. Okay, so cytoplasm means cytosol plus the organelles, but we will not consider the plasma membrane and the nucleus. I hope it is clear to you. Okay, so as you can see the diagram, yeah, again I'm telling you that without the nucleus and the plasma membrane, the liquid portion and the organelle which are present, that is known as cytoplasm. Okay, and cytosol means only the liquid portion. Okay. Now, can you tell me one thing? From where does the cell get the energy? Obviously, there should be some source from where the cell is getting the energy, right? So, one powerhouse is actually present, which helps to give energy to the cell. So I am drawing two. You can also draw more than that. So this powerhouse is actually double membraned, and this is also known as what? This is also known as mitochondria. So let me level that. This is known as mitochondria, and this is also double membraned, and it is known as the powerhouse of the cell, since it produces the energy required by the cell. So let me give the description about this mitochondria. So I will take another page once again and just see this mitochondria is what? Mitochondria is actually made up of two parts, and the meaning of the two parts is that mito means thread, okay, and chondria, or you can say chondrion. This chondrion means granules or grain. Chondrion means granules or grain, okay, or you can say small grains, grain like structure, okay. And what is the main function mitochondria? Mitochondria actually helps in the process of respiration. Okay, so mitochondria actually helps in the process of respiration. So let me write that. So it helps in the process of respiration. You all know what respiration is. Respiration means the breakdown of the food particles in order to release energy. Okay, as you all know that the production of energy by breaking down the food is known as respiration, and this energy is being produced by in the form of ATP, okay. And what is the meaning of ATP? ATP means adenosine triphosphate. There is a full form of ATP is adenosine triphosphate. So these are the energy packets, and in this form the energy is being produced inside the cell by the mitochondria organelle. Okay, that's why mitochondria is known as the powerhouse of the cell. Now I will discuss about the next portion, that is centriole. Centriole actually it helps in the process of cell division. Okay, now I will first draw the centriole. Actually, in order to construct the centriole, you have to put two cylindrical pieces at 90-degree angle, and in this way you have to fit inside the circle. Okay, and as you can see that inside this centriole, inside the cylinders, protein are present, and the name of the protein is tubulin protein. Okay, the name of this protein is tubulin protein. So let me mark it over here, because in that diagram it is very difficult for all of us to mark the tubulin protein. So let me first discuss the function of this centriole. First function I have already said, that cell division, and the second function is that it helps in the production of microtubule and microfilament. Okay, so microtubule and microfilament, which I have already discussed, that it helps to maintain the structure of the cell, and this microtubule and microfilament is actually created by this tubulin protein, and it also helps in the production of the flagella and cilia. Okay, it helps in the production of flagella and cilia. Just see the structure. This is actually flagella, which helps in the movement of the cell, and sometimes you will see this type of hairy structure present around the cell, which is known as cilia. Okay, so for the production of this flagella and cilia, mainly this tubulin protein of the centriole is required. So what you have learnt about the function of the centriole: first, that it helps in cell division; number two, it helps in the production of microtubule and microfilament. Okay, so as I have already discussed about, I will also show you the diagram. Okay, so it helps in the production of microtubule and microfilament, and it helps in the production of flagella and cilia. Now let me bring that diagram once again, and I will draw the centriole with the help of this yellow color. So a small one I will just draw, and then I will level that. You have to level in this way only. Then I will write centriole. Okay, so in this way you have to write centriole. Okay. I hope it is clear to you. Now I will discuss about the next topic, that is, as you can see, this foldings is known as microvilli. Now what is the main function of this microvilli? This microvilli, I will discuss the function, but let me first label this. This is known as microvilli. Okay, so the main function of this microvilli is that it increases the surface area. As a result, whenever it is increasing the surface area, it helps in the absorption of substances. Okay, and it also helps to send outside any substances if it wants to send outside, or the process of secretion is also done by this microvilli. If any substances want to come inside also, it can also do that. Microvilli can do that. Now it also helps the cell to stick to the surface or to attach to other cell. Okay, if it wants to attach to another cell, it can also do that. If it wants to stick to the surface, it can also do that. So this is the main functions of the microvilli, which you have to remember. I hope it is clear to you. Okay, now the next portion, obviously I will discuss, that is about some vesicles I will discuss. So with a different color I will do it. Let me bring a green color so that it will be easier for you to understand. Okay, so microvilli I have completed. So now I will discuss about the secretive vesicle. Suppose any chemicals are produced inside the cell which is to be needed outside the cell. Okay, any chemical is produced inside the cell and it has to be sent outside the cell. So now this chemical is first of all released, and these are actually packed inside the sac like structure. Okay, so let me draw that. These chemicals are enclosed, the chemicals which are released, these are enclosed in a sac like structure called the vesicles. Okay, so let me draw that in this way. These chemicals are surrounded by the this vesicle sac like structure, and the wall of this secretive vesicles is actually made up of what? It is made up of lipoprotein, which I will discuss, but before that let me label this one, that is, this is secretive vesicles. Okay, so this one is secretive vesicles. And now how these chemicals are released outside, let me tell you. So the outside of the secretive vesicle is made up of lipoprotein, and the outside of the plasma membrane is also made up of lipoprotein. So obviously, as you can see, that both are made up of lipoprotein. So whenever they will come in contact, both of them will come in contact, that is secretive vesicle and the cell membrane will come in contact, then what will happen? That will be dissolved, and obviously this chemical will be released outside the cell, just like the way I am marking. Okay, so in this way the chemicals go outside the cell. So this process is known as exocytosis, whenever any chemicals are going outside the cell. Now I will discuss about the endocytosis, that is, if any chemical wants to come inside the cell. So in that case, how the process is being done? So if any chemical from outside wants to enter the cell, they just come in close contact with the cell, and one cut is created in the plasma membrane, and the small chemicals just fit inside that cut. And what happens, the plasma membrane got sealed, and when it got sealed, the one vesicle is being created separately. As you can see, this vesicle is being created, and this type of vesicle which is being created, within which the chemical is present, that is outside chemical is present, that is known as pinocytic vesicle. Let me level that also. This is known as pinocytic pinocytic vesicle. So this is known as pinocytic vesicle. I hope it is clear to you. Now this process is known as endocytosis also. Whenever it is going outside, it is known as exocytosis. Okay, the chemical, whenever it is going outside, the process is known as secretion or also known as exocytosis. And whenever any chemical is entering inside the cell, so that is known as, the vesicle is known as pinocytic vesicle, and the process is known as endocytosis. Okay, so all these organelles you have to remember, because these are the important portions. I hope it is clear to you. Now if you find this video to be helpful, if you find that I have provided enough information about the cell organelles and its part, then don't forget to like and subscribe my channel, and also press the bell icon. Thank you so much for watching the video. Bye bye.